Hey guys, welcome back to my class once again. Today we will discuss about a rare human blood group, the Bombay Blood Group. As the name suggests, this group was first discovered in Bombay, recently known as Mumbai, in the year of 1952. But it's not like that. This kind of phenotype can only be found in Bombay. Rather, it can be found anywhere in the world. And this group is so rare that only 4 individuals are found to bear this kind of blood group among 1 million individuals. So you can imagine the rareness of such kind of blood group. Now this topic is very important for all of us to understand properly. And the topic is also important for class 11 as well as NEET aspirant students. So come, let's start our main discussions. If we want to understand detail about this Bombay Blood Group, then we have to discuss something about ABO Blood Group System. Because this Bombay Blood Group is nothing but an exception to ABO Blood Group System. Although I am not going to discuss detail about ABO Blood Group System in this video. But we all know that according to ABO Blood Group System, there are 4 different types of blood group present in human. Blood group A, B, AB and O, which are actually determined by different alleles of this I gene, capital I gene, which is located on the chromosome number 9 in our nucleus. Now I will not discuss about the genetics of ABO blood group system. Rather, we will discuss about the naming pattern of our blood group system. How this name like A, B, AB or O came into existence. Actually, these names are derived from the presence of antigen on the RBC membrane. Imagine these are different RBC of respective individuals. A blood group, B blood group, AB and O blood group individual. Now the type of antigen that is found on the RBC membrane is actually responsible for determining its blood group, not the antibody that is found in the blood plasma. So suppose in this individual of A blood group, we will find A antigen on the RBC membrane. In case of B individual, we will find B antigen on its RBC membrane. In case of AB individual or the individual that bears AB blood group, we will find both A and B type of antigen on the RBC membrane. And in case of O blood group individual, we will not find any kind of such antigens. Antigen A or B will not found to present on the RBC membrane of O blood group individual. Now the question arises whether this RBC of O blood group individual are actually bald or nude in nature. No, it's not. Rather, it consists of something different kind of antigens. These antigen are actually known as H antigen or H substance. These are actually oligosaccharide in nature, and these H antigens are actually served as a precursor of both A as well as B antigen. Now let's try to find out how these H antigens are actually formed. Imagine this is a RBC of O individual, of such kind of individual. On the RBC cell membrane, there are several oligosaccharide molecules, millions of oligosaccharide molecules are found. Now these structures are present from the beginning. At the early developmental stages of RBC during the erythropoiesis, from a special chromosome number 19, RBC can produce a special type of enzyme known as Foucault's transferase. This particular enzyme actually helps to transfer an additional group on the top of this pre-existing oligosaccharide structures. So Foucault's transferase enzyme helps to transfer a monosaccharide unit known as Foucault's on the top of this pre-existing oligosaccharide structure. So these are the Foucault's molecule. Now this structure together, the fucose and this pre-existing oligosaccharide, these two structures are known to form the H antigen. So the H antigen are actually oligosaccharide structure consisting of fucose and some oligosaccharide molecules. So this is the structure H antigen that is present on the RBC membrane of O blood group individual. Now let's try to find how this pre-existing H antigen serve as the precursor for A antigen and B antigen. Let's start the diagram with the help of a RBC. So this is the RBC of A individual. This RBC also contains some pre-existing oligosaccharides and on the top of this structure it contains the fucose. So these are the structure known as H antigen. Now in case of A blood group, there are some additional structures are found to be attached on the top of these pre-existing H antigen. Let's find what are those structures. In case of A individual, chromosome number 9, here the chromosome number 9 produce a special type of enzyme. The enzyme which is known as N-acetyl galactose amine transferase. This particular enzyme actually help to transfer additional group of N-acetyl galactose amine on the top of these pre-existing H antigens. So these structures are known as A antigen. This entire complex structure contain of oligosaccharide, fucose as well as N-acetyl galactose amine. This entire complex structure is known as A antigen. This is present on the RBC membrane of A individual blood group. Now let's find how B antigens are formed from the pre-existing H antigen. Let's start the same diagram. The surface of RBC, it contains the H antigen. These are the oligosaccharide structure. Oligosaccharide. On the top, these are fucose molecules. Fucose. So up to this, these are the H antigens. Now, in case of B blood group, there are some special type of groups are found to be attached on the top of this H antigen. And the structure is known as galactose. Here, during its erythropoietic development, from the chromosome number 9, these individuals are known to form a special type of enzyme known as galactose transferase. This particular enzyme will then transfer a special monosaccharide unit on the top of this pre-existing H antigen. So these are the complex structure known as B antigen. So these galactose, fucose and oligosaccharide together known as B antigen. So this is how H antigen, being the precursor of A and B antigen, help to produce the A and B antigen in different individuals. And in case of AB individual, they are found to possess both such kind of A and B antigen on their RBC membrane. Now what happens during the Bombay blood group? What happens with the individuals that possess the Bombay blood group? Such individuals are unable to produce such kind of H antigen on their RBC membrane. So their RBC membrane are actually completely bald. They are not able to produce the H antigen even. So the Bombay blood group individual actually contain anti-A, anti-B as well as anti-H antibody in their blood plasma. That's why they cannot receive any blood from any other group other than their own type. So this is the peculiarity of Bombay blood group. So I hope you got clear idea about the topic from our today's discussion. Stay Still, if you have any queries, you can ask me in the comment box below. We will meet again with another important topic in our next video. Till then, goodbye.